Good evening. Good evening, dear students.
Good evening, sir.
What about others? Only 4 members. Just inform to other your friends, Ramadevi and Padmavati. Just inform to your friends. Only 2 members, no? I will wait some more time. Okay.
Okay, sir.
Just inform. Good evening, all of you. Okay, today we will discuss apomixis. So, any idea apomixis? Yes. The first we discuss in the introduction. The normal sexual cycle involves the two important processes. One is the meiosis, another one is the fertilization. So here, the meiosis, this change is the diploid sporophytic cell. It is megasporophyll cell into 4 applied gametophytic cell. These are the meiosis. The meiosis is so many fertilization in which the two haploid gametes of opposite sex fuse re-establish the diploid sporophytic generation. So the sexual cycle, a diploid generation, it is the sporophytic, and the applied generation is the gametophytic. Generally, in angiosperm plants, the gametophyte generation are very short and are represented by the embryo sac. The embryo sac, it is nothing but the female. So, and the microspore, what is the microspore? The microspores, it is also called as the pollen grains. It is male. So, the remaining part of generation, sporophytic generation. In some plants, meiosis and syngamy are interrupted, but still a viable embryo is formed within the confines the seed coat. Such asexual seeds which produce progeny identical for the female parent are called apomycetic seeds, and the phenomena is known as apomixis. Apomixis. Apo means away from, plus mixis means act of mixing or fusion. Yes, do you understand this line? What is the apomixis? What is the meaning of apomixis? The apomixis, apo means away from, mixis is the act of mixing or fusion. So the definition of apomixis defined as a substitution of usual sexual reproduction by a form of asexual reproduction which does not involve the meiosis and syngamy. The substitution of the usual sexual reproduction by a form of sexual reproduction which does not involve meiosis and syngamy. So, in plants with independent gametophytes, for example, ferns, the apomixis refers to the formation of sporophytes by parthenogenesis of gametophytic cells. The apomixis in plants is known as agamospermy. The seeds or embryos are produced by the non-sexual processes. So the plants which independent gametophytes, the apomixis known to occur in more 300 species. It's belonging to the 35 families. Apomixis. The apomixis known to occur more than 300 species. It's belong to the 35 families. It is most common families: Asteraceae, Poaceae, Rosaceae, Rutaceae. These are the common example of apomixis families. So the apomixis is more common in polyploids than in diploids. The apomytic species invariably perennial and often reproduce vegetatively by the stolons or rhizomes. So here, the apomixis derived from two Greek words, apo, away from, and mixis, act of mixing or mingling or fusion. The apomixis is a type of reproduction which is sexual organs of it is related to the structure take part, but the seeds are formed without union of gametes. The first discovery of this phenomena is credited the Leeuwenhoek as the 1719 in the citrus seeds. The genotype of the embryo can resulting the plant will be the same as the seed parent. This is clonal seed production. So here, so these are the modes of reproduction. It is the sexual, asexual. The asexual is vegetative reproduction and apomixis. In apomixis, in the again two types, gametophytic and sporophytic. So the development of embryo without sexual fusion. The sexual life cycle is short circuited. The genotype of developed plants are identical to the parental plant. It is discovered by the Leeuwenhoek in the 1719. So coming to types of apomixis. So the apomixis involve two main categories. One is vegetative reproduction. One is agamospermy. Agamospermy. Like, so here, so many definitions are there. Just write down. The process of development of diploid embryos without fertilization. This is also definition of apomixis. And another definition, apomixis is a form of asexual reproduction. Apomixis is a form of asexual reproduction that occurs via seeds in which embryos develop without fertilization. And apomixis is a type of reproduction in which sexual organs is related to structures take part, but the seeds are formed without union of gametes. So in some species of plants, the embryo develops in the diploid cells of the seeds and not a result of fertilization between the ovule and pollen. This type of reproduction is known as apomixis, and the seedlings produced in this manner are known as apomix. The apomycetic seedlings are identical mother plant, similar plants raised by the vegetative means. So here, coming to types of apomixis. Types of apomixis. So here, according to Maheshwari 1950, it classified apomixis into 4 groups. Recurrent apomixis. In this type of apomixis, the embryo sac develops from the diploid egg mother cell or from the some other diploid cells of the embryo sac without fertilization. What are the concept here? Here, the formation of the embryo sac develops the diploid egg here without fertilization. So the egg has normal diploid number of chromosomes, the same in the mother plant. The embryo subsequently develops from the egg nucleus without fertilization. This series of events is now occurs in the some species of Cripis, Poa, Taraxum, and Allium without stimulus of pollination. In this, like Parthenium and Rubus and Malus, the stimulus of pollination appears to be necessary for the development of embryo or to produce the viable endosperm. These are the recurrent apomixis, non-recurrent apomixis. So, in this type apomixis, the embryo develops directly from the applied egg cell, applied parthenogenesis, and some other applied cells of embryo sac. It is applied agogamy. Without fertilization, as a result, the embryo developed is also applied. This type of apomixis occurs very rarely and is primarily a genetic insert. The plants produced are applied and sterile. It is common in Solanum niagrum and lithium species. So these are the non-recurrent apomixis. Next, advantageous embryony or nucellular embryony. So advantageous embryony is also called as nucellular embryony or nucellular budding. In this type of embryo does not develop from the cells of the embryo sac, but it develops from a cell or a group of cells, either nucellus or integuments. The cells integument or nucellus are diploid, and hence the results of embryo is also diploid. Such embryo usually develop outside of the embryo sac in the addition of the regular embryo. The advantageous embryony occurs in the many plant species, but the most prominent tropical and subtropical tree species like citrus and mango. So next, vegetative apomixis or bulbis. In this type, the vegetative buds or bulbis are produced in the inflorescence in place of the flowers. These buds or bulbis may support into new plants while they are still attached to the mother plants. This type of apomixis is quite common in Oleum and Agave, Poa and Dioscorea and in some of the grasses. So here another one is type is not mentioned. Angiosperms. These plants retain this type not in the slide. So this plant retain the seeds of agent propagation, but the embryo is formed by some process in which normal meiosis and syngamy have been eliminated. So Angiosperms includes two different types, gametophytic and sporophytic or adventive embryony. So here, gametophytic apomixis. So the embryo sac develops from a diploid megaspore mother cell without meiosis directly from the A cell in the nucellus apospory. The diploid egg develops into an embryo without the participation of the male nucleus. The formation of embryo from an unfertilized egg is called parthenogenesis. These are the gametophytic apomixis. And here, again, these gametophytic apomixis in this two types, diplospory. The megaspore mother cell differentiated into sexual ovules, but does not enter meiosis. It produces diploid embryo sac through the mitotic divisions. Example is Taraxacum, Lexericus, Alternaria allium. And second one is apospory. This is the most common mechanism apomixis in the higher plants. It was first reported by Rosenberg, 19th century. In this normal megaspore mother cell differentiate in the same manner in the sexual ovules wings to enter into the meiosis. In the diplospory, not enter into the meiosis. The division may not completed to form a linear tetrad. At some stage during this period, one or more adjacent similar cells become a meristematic and appear like megaspore mother cell. These cells undergo mitotic divisions to form unreduced embryo sacs. The sexual megaspore mother cell are its products about the aposporous embryo sac occupies the entire area of the oval. The unfertilized eggs of such embryo sacs gives rise to the diploid embryos. So, apospary, it is mainly two types, Herasium type and Panicum type. So, Herasium type, the megaspore mother cell undergoes the usual meiotic divisions and form a tetrad. At this stage, a nucellular cell at the chalazal end of the tetrad becomes activated and starts developing into a aposporous, unreduced embryo sac. All the four megaspores degenerate and the aposporic embryo sac is matures. So, panicum type. The aposporous embryo sac is four nucleated with three-celled egg apparatus. It is a polar nucleus. Antipodals are absent. Example is panicum, poa, pennisetum. These are the regarding about agamospermy. So, advantage of apomixis or significance of apomixis. The assured reproduction is absence of pollination such as extreme environments. The maternal energy not wasted in the unfit of offsprings. Some apomictic plants, but not all, avoid the male energy cost of producing pollen. And the apomixis do not involve the meiosis. Hence, the segregation and recombination of chromosomes do not occur. The apomixis helps in the preventing the desirable characters of the indefinite periods. The apomixis simplifies commercial hybrid seed production because isolation would be necessary to produce F1, the maintain the multiple parental germination. The apomix is not very common in the major crop plants. Inspired by the So, my voice is audible. Many disturbances.
Yes, sir.
So, these are the some useful advantages or significance of the apomixis. So, the disadvantage can't control the accumulation of the deleterious generic mutations, usually restricted to the narrow ecological niches, lack ability to adapt to the changing environments. So, what are the next topic? Just you see the your textbook. So these are the regarding apomixis. I think this question mostly the short answer, not a essay answer. Mostly. So next polyembryony, right? Wait. So coming to polyembryony. So what is the polyembryony? Yes.
More than one embryo.
One embryo is small.
Okay. Before that, the after fertilization, the ovule mature into the seeds. Yes, after fertilization. Generally, the ovules it is becoming or converted into the seeds, right? After fertilization. So mature into the seeds. The after fertilization, the ovule mature ovules into the seeds, and the zygote develops into an embryo. Okay, already we discussed in the previously also how to formation of like embryo. We discussed in the development of embryo already we discussed. But after fertilization, the mature ovule it is converted or it is seed, like yes, and the zygotes develops into an embryo. And normally a single embryo is present in the each seeds, right? And single embryo, do you catch the my word? Single embryo is present in the each seed, right? Yes, generally, this is generally single embryo present in each seed. But sometimes more than one embryo may be present. More than one embryo present in a seed, it is called as polyembryony. Poly means many, more than one. Poly means many. So, one embryo may be present in the seeds. This is a general condition. Okay. But sometimes more than one embryo present in the seeds, this condition known as polyembryony, right?
Yes.
The mature seed contains only one mature embryo. Strictly speaking, a polyembryony includes all such instances where there actual occurrence of two or more embryos in the developing ovule, except for a few taxa, that is citrus mangifera. The polyembryony occurs in the abnormal feature. So polyembryony was first time reported by Antony van Leeuwenhoek in the 1719 in the seeds of orange. Since then, it has been observed in the large number of tags of angiosperms. Firstly, this phenomena was initially discovered by Antony van Leeuwenhoek, the 1790. Okay, do you clear what is the polyembryony?
One embryo is
more than one embryo present in the seed.
More than one embryo present in the seeds known as polyembryony. Okay, so the polyembryony is common among the conifers. It is the gymnosperms, but many species of the both dicotyledons and monocotyledons exhibit this phenomena. So the polyembryon we are seeing in the dicotyledons and monocotyledons, and common in the gymnosperms, right? So classification of this one. So the polyembryony is two types: spontaneous and induced. So the spontaneous includes instances of naturally occurring polyembryony. Induces instances of the experimentally induced in the polyembryony. So spontaneous polyembryony has been subdivided by Ernest Saint in the 1918 into two categories. This was also adopted by other leading of embryologists, according to the Maheshwari Sharnoff, the scientist Maheshwari 1950. and Chernoff 1929. First one is that true polyembryony. What is true polyembryony? So here, the true polyembryony, again, it is subdivided into two types. The first one is cleavage polyembryony, adventive polyembryony, right? So first of all, true polyembryony. So the production of embryos within or by projecting into a single embryo sac is termed true polyembryony. What is the meaning of the production of embryos? Production of embryos are by projecting into a single embryo sac. The production of embryos within the embryo sac, the term is called as true polyembryony. Got it? These are the regarding diagrams. And cleavage polyembryony. Cleavage polyembryony. So where the embryos arise within an embryo sac, either by cleavage of egg or from the synergid antipolars or endosperms. The cleavage polyembryon confers this is the normal embryo. And this the below is the suspensor. The lateral divisions. Lateral divisions is nilu. These are vertical means this type. The cleavage lengthways, it is the new embryo. This is the cleavage polyembryony. So next one is adventitious polyembryony. Adventitious polyembryony. The embryos arise from the tissue laying outside of the embryo sac. Outside of the embryo sac, this is the normal embryo, the nucellular embryo. These are the endosperm, this is the integument. So this is example of the citrus oval young seed. In the section showing in the normal and nucellular, it is nothing but adventive embryos. So the cells of the nucellus are integuments, but generally they come to lie within the embryo sac. So next, false polyembryony. False polyembryony. This type includes the cases in which two or more embryos are formed as a result of the development of the aposporic embryo sac. Aposporic embryo sac, right? So next, origin of embryo from the synergids or antipodal cells. The embryos may also be produced from the other parts of the embryo sac, such as synergid. The synergids and antipod also, these two cell operation also, the capacity it is produced embryos. Not only embryo sac, the synergids and antipodal cells also produce the embryos. So that's why the origin of embryos from the synergids are antipodal cells. These are the zygotic embryo, these are the antipodal embryo. The polyembryony, the formation of embryo from the antipodal cells. Here. So in most cases, synergids become a egg-like to form the embryos with or without fertilization. As well as antipodals. Antipodals produce the embryos, and the synergids also produce the embryos with or without fertilization. The production of embryos from antipodal cells is rare. The production of embryos from the antipodal cells is very rare. So these are the zygotic embryo. It is the synergetic embryo. This synergetic embryo produce the embryos. So polyembryony formation of synergid embryo in the ileum. Okay. So next, origin of embryos from the endosperms. Endosperm also produce the capacity of the produce the embryo. So tube in the 1898 in the Balanophora, Woodworth, the Alnus and others have reported the embryos developed from the endosperm. The embryo develops normally from the egg. The origin of embryos from the cell outside nucleus are different. The embryos also develop from the cells of the nucellus and integuments. Not only synergids, antipodals, the embryos from the nucellus and integuments also.
Page number, sir.
Just wait. Ramya, you want page number? So the page number is
248, sir.
yes, 248. Ramya, the page number is 248. Thank you. Next, these are the different process and different productions of the embryos in the different cells, embryo sac cells. The origin of embryos from the other embryo sac in the ovule. So here, the origin of embryos from the cell outside the embryo sac. In many parts, the production of the embryos. Here, the causes of polyembryony. Many theories have been proposed to explain the occurrence of polyembryony by different workers in the different times. Some of the important theories are as follows. Necrohormone theory. This theory indicates that the degenerating cells of the nucellus act as the source of stimulus for the adjacent cells to divide and form adventive embryos. So hybridization theory. Next theory is hybridization theory. According to this theory, the occurrence of the multiple embryo is due to the hybridization. According to this theory, the occurrence of multiple embryos, why it is formed in the multiple embryos, it is due to the hybridization. The recommendation of genes takes place during the process of hybridization, forming of a single unit that gives rise to multinucleate embryos. So next, coming to the importance of the polyembryony. So plant breeding, it is plant breeding in the horticulture. Nucellular embryos are supposed to be free from the disease. So propagation of the fruit trees such as the citrus and mango. The application of adventive embryos is also important for providing genetically uniform. The seedlings in the fruit trees can be used for the development of homozygous diploid. The artificial production of these embryos from the eggs are synergids. So these are the regarding about polyembryony. So these are the mostly two short answers, maybe important in this chapter: apomixis and polyembryony. Most important in the apomixis. So next we jump to the embryology of angiosperms in the parthenocarpy. So, you know parthenocarpy? Just a minute.
Production of foods without fertilization.
Without fertilization. Without fertilization means without
Formation
seeds.
of zygote. Without formation of zygote, I think.
Yes.
Fruits without seeds.
Just wait. Just wait. Okay, just wait. I will drink some water. So, all of you, parthenocarpy. So, what is the definition of parthenocarpy? The fruits are created without the need for ovule fertilization. Without commonly, the definition is without fertilization. So, the parthenocarpic fruits lack of endosperms and embryo. So, what happening? There is no endosperm. There is no embryo. Next, what happening? There is no seeds. Yes, generally the parthenocarpic fruits called as seedless fruits, right? Most of the hybrid varieties are the parthenocarpic fruits because the hybrids there is no seeds. Like in the streets, you observe some big size of guavas are there. So that guavas compared to in the local guavas, there is no not much more seeds. Only the little bit of seeds are there in the guava, hybrid guava. So most of the hybrid fruits there is no seeds. So the definition is, yes, tell me what is the definition?
Fruits without seeds.
Yes, so everyone knows. So the producer, this was initially presented in the 19th century. The gibberellic acid is a plant hormone that is introduced together with parthenocarpic. The plants are beginning grown. The gibberellic acid is present in the parthenocarpic seeds. The parthenocarpic fruits. This causes the ovary to mature without being the fertilized, producing large pulpy fruits. In consequence, the examples of the parthenocarpic fruits includes banana, pineapple, watermelons, and cucumbers also. Sometimes apples, so many tomatoes. So, the types of parthenocarpy. There are three main subcategories of the parthenocarpy, vegetative parthenocarpy, stimulative parthenocarpy, and stenospermocarpy. So first of all, we go for the vegetative parthenocarpy. So without pollination, vegetative parthenocarpy definition is without pollination, this kind of parthenocarpy results in no seed development within the seeds. So the pears and figs are two example fruits that have it. These are the vegetative parthenocarpy. Without pollination, so what happening? Without pollination, the parthenocarpy results is no seed development within the seeds. So pears and figs are two examples of the fruits that have. So next one is stimulative parthenocarpy. So although there is no pollination, here also there is no pollination, there is use of an external stimuli. The external stimuli, this can be a wax ovipositor placed into the flower of ovary, or use of plant growth regulators and the unisexual blooms found inside of the syconium. So syconium is a line of the unisexual flower with a flask-shaped structure. So it is the stimulative. So any stimulator. So this type also there is no pollination. Next, stenospermocarpy. Stenospermocarpy. This particular kind of parthenocarpy is distinctive that all the fertilizations occur and the seed starts to grow. It ultimately aborts within a fruit. The trace of the seed may be seen, indicating the location where seed development ended. The fruits like watermelons and seedless grapes exhibit this sort of the parthenocarpy. The fruit without seeds are bred to take advantage of immature seeds before they are aborted. These partially formed seeds are taken out of the fruit and used in the tissue culture methods to grow into the plants. Both parents inherit the seedless characteristics, which helps to produce a large number of the seedless offsprings. So, explanation of the parthenocarpy with an example. The following over the seedless watermelon are examples. So, how to produce the seedless watermelons? They are giving some example. The parthenocarpy occurs in the stenospermocarpy. The numerous factors contribute to the stenospermocarpy. One of the process of 3 chromosomal sets, while most organisms have the even number of chromosomes. Seedless watermelons contain 3 sets of generally even number of chromosomes, like 2, 4, 6. But here watermelon, it contains 3 sets of chromosomes in the each cells, resulting in the pollen and egg cells receiving the same. These cells, often referred to triploids, do not get an equal number of chromosomes as they develop into pollen and egg cells. This leads into the abortion of seeds development, since knowledge required to generate a viable cells is not present. So triploid organisms can be created either naturally or by coming to tetraploid cell with a diploid cell to create triploid cell. Due to the poor seed development of triploid, the pollens of the fact they cannot germinate diploid cultivars are interplanted to the promote fruit output. It remains the seeds may be clearly observed in the seedless watermelons. Additionally, it should be noted the stenospermocarpy or fruit without seeds, it caused by the naturally occurring point mutation in the region of the grape chromosome that control the seed production. So the parthenocarpy offers benefits and drawbacks. So advantage, the parthenocarp particularly it is well liked by horticulturists fruit beverages, jams, and sauces are more frequently made from the seedless fruits. Fruits freshly portioned grows at the results of this process. The procedure also enables a planter to prevent the insects and pests from the damaging crops without use pesticides. Plants can be covered to stop dangerous insects from the harming of the crops. Pollination insects are not required. So disadvantage. The plant and fruit yields can also harm by the parthenocarpy when it is artificially induced. As an instance, early administration of the phytohormones like auxins prior to the blooming stage or the opening the flower bud, so can harm the flowers, resulting in the abortion of the seeds and fruit drop. So benefits of the parthenocarpy. So it yields fleshy, seedless fruits. It lowers expenses of the cultivation. With the usage of the pesticides, crop productivity can be increased. Larger fruits are produced with the use of the natural plant growth regulators. Reduces the applications of chemical pesticide to fend against the hazards, insects in the infestations. The natural and artificial parthenocarpy. An example of natural parthenocarpy when it develops the results of male sterility, unfavorable environmental circumstances, a lack of pollinators. Parthenocarpy performed using the artificial methods referred to the artificial parthenocarpy. So irradiated pollen and the spraying of the plant development hormones like auxins, gibberellins, which aid in the growth of the fruits, can be both be used to create the artificial parthenocarpy. So this is the regarding about parthenocarpy. So these are the benefits. It produces seedless, fleshy fruit, reduces cost to cultivation. It helps the increase of crop yield with the use of pesticides. Natural plant growth regulators are used, which help up in the production of larger fruits. Reduce the uses of the chemical pesticides, prevent the attacks from the harmful insects. So that is parthenocarpy. What is the parthenogenesis? What is the parthenogenesis? So the parthenogenesis is defined as a process in which a new individual develops without fertilization and ovum. It takes place in the invertebrates and some lower plants. In this process, male and female gametes are formed, but they do not fuse, and therefore it is also called incomplete sexual or asexual reproduction. So the offspring of applied and a clone, a female, the offspring cannot reproduce sexually. The parthenogenesis occurs naturally in the queen bees. The unfertilized egg of queen bee develops to form a male drones having a haploid set of chromosomes. The parthenogenesis can also be done artificially in mammals in the produce of the identical offspring.
Hello, Anna, namaste.
So next we will discuss significance of the parthenogenesis. The parthenogenesis is an important process due to the following these reasons. It can help to know the sex an individual. For the example, honeybees, wasps. The process helps newly formed offspring. This process can help to eliminate the variations from the population. So it has easy and stable process of the reproduction. It can be used to develop a beneficial mutant characteristics of organisms. It can help to control a non-adaptive combination of genes. So difference between the parthenocarpy and parthenogenesis. Parthenocarpy and parthenogenesis are the alternative form of the reproduction that occur without fertilization. Parthenocarpy helps in the formation of fruits without seeds, and the parthenogenesis is a process in which an fertilization ovum forms the new individual. The new individual in a clone of the female and it is mostly haploid. So you can, the difference between the parthenocarpy and parthenogenesis from the table given below. So these are the table, parthenocarpy and parthenogenesis. It is the process of fruit development without fertilization of seeds in the plants. So in this process, a development of a new organisms without fertilization and the ovum. This process lead to the formation of seedless fruit. The organisms produced by the process are a clone of female. They cannot reproduce sexually, cannot produce the offsprings. Only haploid offsprings can be produced. So it occurs only in plants. It occurs in the invertebrate animals and the lower plants. So example is cucumber, banana, grapes. Example, the insects and lizards. So these are the regarding about parthenogenesis and parthenocarpy. Okay. So these are the more some advantages. So parthenocarpy also in the exams, in theory exams, asking in the short answers. What are the, just you write the what is the parthenocarpy, types of parthenocarpy, then you directly go for the benefits of parthenocarpy. So this is more healthy and results are achieved easily, provides seedless fruits and improves quality. It reduces the complete cost of the cultivation. This improves crop yield without using the organic pesticides. The plant growth regulators are natural and it improves the crop yield without using the organic pesticides. Plant growth regulators are natural and the fruits produced are the larger. The parthenocarpy keeps the insects and the pests away without using the chemical because there is no requirement of pollinating insects for the formation of fruits. So this protects the plants from the being attacked by the pesticides. These are the benefits of the parthenocarpy. So, so next class we will say. So next class we will discuss in the next chapter. Next chapter is experimental embryology and application of embryology in taxonomy and agriculture and horticulture. So principles and application of the paleontology. So these three remaining.
Yes, sir.
So how many classes? 3 classes are there, right? So here in this unit, the long question is mostly the parthenocarpy is the short questions. The application of embryology in taxonomy, agriculture, and horticulture is frequently they are asking the question in the your theory exam. Okay. So here, what are the some taxonomy? How it is embryology? It is used for the taxonomy. Horticulture, how they are used in the embryology in the horticulture. So the application of embryology in the agriculture. Okay. So these are the some important question regarding to your exams. And in this unit, just wait. So and another question important in the fifth block 5th, the unit 20, principles and application of the paleontology. What is the paleontology? Any idea? Paleontology.
Study of pollen grains.
Study of pollen grains.
Study of pollen grains. Okay. So you know pollen grains, what are the types of pollen grains? What are the structure of pollen grains? In the different species, in the different pollen grains are there. So these, I think 5 pollen grains and in your practical syllabus, like acacia, hibiscus. So just you already the permanent slides are there. Just you observe the under the microscope, you identify the which pollen grain it is represented, like hibiscus pollen grain or acacia pollen grain, whatever may be, just you identify. Then you will write the some 6 to 8 points regarding that. And also one more in the major experiment in this chapter, the isolation of the pollen grains. Some technology I forgot it. Some technology. Some we are using some technology. So that is also some blue technique something. So that is also one major practical and your practical examination. How to isolate? How to isolate the pollen grains? So this is the today class. Just we remaining in the 3 chapters: experimental biology and application of embryology in taxonomy, agriculture and horticulture, and principles and applications of the paleontology. So this is very simple, very simple and definite question. Simply, parthenocarpy means development of fruit without fertilization or seedless fruits, right? Yes.
Yes,
For
sir.
example, they are giving some example. Apple is a false fruit because it develops from the floral parts. Apple is a false fruit because it develops from the floral parts of the apple tree. You know this sentence. Apple is a false fruit because it develops from the floral part of the apple tree, and banana is a parthenocarpic fruit because it develops without fertilization. So parthenocarpic fruits are those which develop without fertilization. The parthenocarpy helps in the getting seedless fruits from the desired plant or tree. So, and most animals procreate through the parthenogenesis, are small invertebrates such as bees, wasps, ants, and aphids, which can alternate between the sexual and asexual reproduction. Parthenogenesis has been observed in the more than 80 vertebrate species, about half of the are fish or lizards. Parthenogenesis, parthenos, virgin, and genesis is the creation, is a natural form of asexual reproduction in which uh, growth and development of embryos occurs without fertilization by sperm. An animal's parthenogenesis means development of an embryo from unfertilized egg cell. In plants, parthenogenesis is a component process of the apomixis. So here the parthenocarpy is the production of fruits without fertilization of ovals. Fruits like banana and figs are developed without fertilizers and do not produce any viable seeds. So the parthenocarpic fruits example: pineapple, banana, cucumber, grapes, watermelon, orange, grapefruit, pear, fig are the parthenocarpic fruits. These are the developed without fertilization, are often seedless also. So these are the types already we discussed previously. So in the 4th unit, embryology of angiosperms. In this, mostly the apomixis is the short answer. So in development of endosperm or development of embryo, in this, out of these, one long question definitely. Unit 13, 14, compulsory one long question. And development of endosperms or development of embryo. Out of this, compulsory one question. And the short answer is apomixis. So here the biology of angiosperms, parthenocarpy is the guarantee question. So just you prepare principles and applications of the paleontology. These are the most important questions. So sometimes ask embryo applications of embryology in the taxonomy, agriculture, and horticulture. It is very less chances, but principles and application of the paleontology. So this one the new chapter. This one the new topic. So compulsory they give this question, paleontology question. So in this unit you prepare in the short answer parthenocarpy, and the long answer is the principles and applications of paleontology. So block 3. Here, so compulsory in this structure of oval And types of ovules are very important. So already I told you. So either development of male gametophyte, either development of female gametophyte. It is the compulsory question in the long questions. So fertilization is the short answers. Okay. Sometimes they are asking sexual incompatibility. So here the unit 9 and 10 is very, very important. The essay question. So here unit 2. Yes, my voice is audible.
Yes, sir.
Unit 2. Sorry, black 2. Unit 6 and 7 is very, very important. Unit 6 and 7. What are the unit 6? Pharmacognosy and adulteration of the plant drugs, and another one is the
Ethnobotany, history, scope and importance.
Importance.
And
Right?
importance.
Yes. And coming to black 1. So already I gave the some morphology and active principle of medicinal value 1 and medicinal value 2. Some of the important medicinal plants are there according to your syllabus. So they give the compulsory out of, I think, the medicinal value of 2. How many plants are there? 3 or 4. That 2 or 3. So out of 3, 5, one long question is compulsory. So and another question is, so like they are saying, what is the morphology active principle of the Gymnema sylvestre or Vithamia somnifera or Rauwolfia serpentina? This type of questions. Okay. So and the phytochemistry and medicinal plants. These are also very, very important. So these are the important questions. Next, any doubts regarding these important questions?
Sir, phytochemistry essay question, sir.
No. Phytochemistry, just wait. In the many topics are there, the phytochemistry. So it is, you prepare in the phytochemistry. So it is better prepare in the essay question. It is cover both the questions. It is short answer covered and long answer covered. This is the general topic actually, phytochemistry.
Yes.
So compulsory in the long question, the unit 3 and 4. Morphology and active principle of medicinal value 1 and medicinal value 2. This, in the textbook, they given some examples. Out of this, you compulsory one question. And also the phytochemistry also you prepare entire chapter. You will get easily short answer and long answer also. So next. Yes.
Sir, can you send important questions, please, now?
So last day of class, we will. I tell to all of, because in the last class 27th, just you, I requested to all of you compulsory attend the last class. So I give the some important questions that day. Okay, the unit wise.
Thank you, sir.
That's better.
Okay, sir.
Because some of the people are missing, na.
Thank you, sir.
Thank you, sir.
So,
Sir, conservation of medicinal plants, sir.
So that is general topic. So there is no preparation of that, because everyone knows conservation of medicinal, how to conserve the medicinal plants is general topic. So everyone write the general, that is the general topic. So that's why some good knowledge of you. Conservation, how to protect the medicinal plants. It is very simple. Gardens, herbal garden, you prepare a herbal garden. So many, like, just once look this topic, it is easily for writing for your exam. So only I focus in the guarantee questions, but remaining you also just one you look the one read also. That's better. Don't depend only my questions.
Yes, sir.
You all of you just you taken from the Kumar sir, from other sirs, from the old papers how to take question forms. Like one unit suppose block one is there, the block one too long one short, or too long too short. Then what are the questions in frequently they are asking? Just you observe it. It is easily find out how to prepare in the exam. Okay. Thank you.
Okay, sir. Thank you.
Okay, sir. Thank you.
Okay.
Thank you.